Hello students, how are you all? Welcome to One Page Biology. In today's video, I will be covering a very important topic which is called as DNA replication. Now dear student, this topic is from the chapter Molecular Basis of Inheritance. And mind me dear students, Molecular Basis of Inheritance has got a lot of weightage in your board exams as well as in your NEET. So if you want to screen dear students, it has been written that DNA molecule, as in the background you can see there's an image of DNA which is rotating. So please remember dear students that DNA is a double-stranded structure, and when I say double-stranded it means it has got two strands, and the two strands are majorly they are arranged in a helical manner, and at the same time basically both the strands are anti-parallel in nature. So I will be explaining you that in the coming part as well. So what has been written over here is, as you can see on the screen, that DNA molecule, it regulates and controls the cell activities. So as we all know that DNA is the major molecule of every cell. It is present in the nucleus, it is the genetic material, and it definitely controls the entire cell. That means it controls the formation of proteins, it controls the formation of RNA, and without DNA actually the cell cannot survive, right? So it is the most important molecule which is present in the cell. So let's proceed. We are going to talk about how the DNA molecule divides or how it undergoes replication. So now as you can see on the screen dear students, that DNA has got two important functions majorly, which one is called as a heterocatalytic function. So here the word heterocatalytic, what do you mean by the word heterocatalytic? So here hetero means different. As you all know the word in biology, always remember the word hetero means different. So in this case it means that DNA, as we said, it helps in the formation of other molecules. Something like, for example, it helps in the formation of RNA, whereas it also helps in the formation of proteins, whereas proteins are formed from RNA only. So when the DNA helps in the formation of proteins, which is a different molecule, or to be specific, when it helps in the formation of RNA, which is a different molecule, such a function will be considered as, come on dear students, such a function will be considered as a heterocatalytic function. Right? So as you can see in this particular picture, from DNA what is formed is RNA, and from RNA what is formed where the RNA helps in the formation of protein. So from DNA, when the RNA is formed, such a process is called transcription, which is a different process altogether. There is a mechanism which is there. I will be trying to make a video on transcription also. And from the RNA helps in the formation of protein, and this is by the process of translation. So this is also a different process, dear students. So when the DNA helps in the formation of RNA, and from RNA what ultimately is formed is proteins, this can be considered as a heterocatalytic function of DNA. Whereas if you talk about DNA, which can form DNA on its own, like from DNA only its own formation occurs. The new DNA is formed from the parent DNA, and this is by the process of something called as DNA replication, and that's what we are going to today understand. So here replication will be considered as an autocatalytic function, dear students, right? So as you can see here in this picture, as I said, DNA can form its own copy, or you can say it can divide on its own. So this will be considered as a autocatalytic function. So we said DNA has two functions, heterocatalytic, in that it means it forms RNA, which is a different molecule, and when we say DNA performs autocatalytic function, means it can form its own molecule, that is by something called as DNA replication. Okay. Now let us understand, dear students, who actually came out with this process of DNA replication, or who actually found out this process of replication. So dear students, you all must be having an idea about this, that this process of DNA replication was found out by two very famous scientists, and their name was Watson and Crick, dear students, because they were also known for finding out the structure of DNA, which we all today know it as a double helical model, right? So please remember their name. Watson and Crick actually found out the process of replication. They have constructed a 3D diagram of DNA, and as you can see here, there is Watson and the other one is Crick, who is actually studying this entire DNA model. And such a model which they had made, this model is called as a semi-conservative replication model. Now why it is called semi-conservative, that you will understand when we completely understand the process of replication. I'm sure at the end you will understand why it is called semi-conservative. But please also remember what is being said over here is it is based on this model which they had explained, is based on the principle of anti-parallel and complementary nature of DNA strands. What does it mean, dear students? It means that the two strands which are present in the DNA, they are actually anti-parallel in nature. Anti-parallel means they are in the opposite directions, they are not in the same directions. At the same time, they are complementary to each other. So what is complementary, that also we will see in the coming part. So let us start by understanding how the replication starts. So for that we need to understand in a stepwise manner. So let us understand, dear students, the first step of DNA replication. So what is the first step? The first step is called as activation of nucleotides. Now try to understand, dear students, as we said that DNA is two-stranded structure or double-stranded structure, and for the formation of new strands, what is required is nucleotides. What are nucleotides? Nucleotides are, you can consider it as the monomers or the units from which the DNA strand is formed. Correct? So for the formation of new strands, obviously there is a need for fresh nucleotides or new nucleotides. And how are these nucleotides helping actually? So first of all, the nucleotides which are there on the left hand side, you can see here, there are four basic nucleotides which are involved in DNA replication or which are involved in the formation of DNA. These are DAMP, DCMP, DGMP and DTMP. What is DAMP? DAMP stands for deoxyadenosine monophosphate. Right? So likewise, see here, DCMP will be deoxycytosine monophosphate, and DGMP will be deoxyguanosine monophosphate and deoxythymosine monophosphate. Now all these are basically nothing but nucleotides, but directly they cannot be used in the formation of strands. So what they need to do is they have to be activated, my dear students. And when I say activated, that means they need to be filled up with energy. And who is going to give them the energy? Yes, my dear students, you must have guessed it right. It is ATP molecule, ATP. So every nucleotide will combine with ATP to form a highly energetic nucleotide. For example, DAMP, when it combines with ATP, it forms DATP. When DCMP combines with ATP, it forms DCTP. DGMP combines with ATP to form DGTP, and DTMP combines with ATP to form DTTP, wherein DATP is nothing but deoxyadenosine triphosphate, right? DCTP will be deoxycytosine triphosphate, DGTP will be deoxyguanosine triphosphate, and DTTP will be deoxythymosine triphosphate. Now you will be wondering, where exactly is this occurring? Like, where are the nucleotides getting charged up or where are they getting activated? So these are getting activated inside the nucleus itself. And as you all know, the fluid inside the nucleus is known as you can see on the screen, it is called as nucleoplasm. Right? And this entire process, as you can see that the nucleotides are getting activated with the help of ATP, this is carried out by the enzyme phosphorylase, and the process is known as phosphorylation, because obviously there is addition of phosphate, and who is giving the phosphate? ATP is giving the phosphate. Right? So let's proceed. So once the nucleotides are activated, now next is basically, now once the nucleotides are activated, the DNA replication process will start. So this start of DNA replication is known as initiation of DNA replication, or you can say initiation means beginning of DNA replication. So how does it start? So it specifically starts at a point on the DNA, dear students. So as you can see in this particular diagram, there is a particular point on the DNA. Now this is a case of prokaryotes. Prokaryotes majorly means basically what is included in prokaryotes are the bacteria, right? Whereas the eukaryotes are the well-developed cells which have majorly a well-developed nucleus. So So you can see here both in case of prokaryotes and eukaryotes. In case of prokaryotes, there is a point from where the DNA replication starts, whereas in eukaryotes also there is a point where the DNA replication actually starts. So this point is known as origin of replication, right? Here they are saying that here also there is origin of replication. But what is the difference between prokaryotes and eukaryotes? As you can see on the screen, in prokaryote there is only a single point where in this entire DNA, as you can see, this is a circular DNA in case of prokaryotes. So there is a single point of origin of replication. There is only a single point. But in eukaryotic DNA, there are many points where actually the DNA will start the replication, and that's why it is said that it has got multiple origins of replication. Okay. At the same time, the point where now, as you can see here, the replication is begin, right? So there will be a point where basically the replication will stop, and that point is called as termination point, or you can also call it as the termination site, right? So you can see in this particular diagram, as you can see here, dear students, that this is a point. From here the replication has begun, but at the same time this region also has something called as termination site. So as soon as the DNA has been replicated, the new strands have been formed, the DNA replication will stop at this particular point, which is known as the termination site. Now this entire part wherein the DNA is getting replicated, right, from the point where it is starting and the point till the point where it ends, this entire part of replication is known as unit of replication, and it is also known as replicon. What is the unit of replication called as, dear students? It is called as replicon. Fine. Now this is a point, you can consider it as the origin. So at this point there is a small nick which is created. What do we mean by a nick? A nick is nothing but a small break which is created. That means here there is some bond which is there, which connects the nucleotides, right? So how is a nick created? This nick is created by breakdown of the phosphodiester bond, dear students. So as at this point, which bond is present? This bond is known as a phosphodiester bond. And what is a phosphodiester bond? Phosphodiester bond connects the neighboring nucleotides. As you can see, the nucleotides, these are connected by phosphodiester bond. So at the origin of replication, as the DNA replication begins, this phosphodiester bond will be broken, so that now the strand is actually going to break at that point. Now followed by, once the nick has been created, now what will happen is, in the third step there is this particular enzyme which is known as DNA helicase. This is a very important enzyme, dear students, because this enzyme is going to separate the two strands, and that's why it is called as unwinding of DNA molecule. What do we mean by unwinding of DNA molecule? That means the two strands are going to separate from each other. And how are they going to separate from each other? What are the bonds which actually join the two strands? These bonds are nothing but the hydrogen bonds. So this particular enzyme, that is DNA helicase, is going to break the hydrogen bonds, and that's how it is going to separate the two strands from each other. And as you can see in this picture, that the two strands have started separating from each other. So this helicase enzyme will break the hydrogen bonds between the two strands, and as a result of which the two strands will get separated, and this process is known as unwinding of DNA molecule. Now once the two strands actually separate, you can see this particular shape of the DNA. Now this shape of DNA is looking like something called as a fork, and that's why this is known as a replication fork. It is also considered as a Y-shaped replication fork because this is an inverted Y-shaped structure, and that's why it is called as Y-shaped replication fork. Unwinding is bidirectional. That means from both the sides, dear students, the replication fork from both the sides, that is from this point as well as from this point, the DNA is unwinding. So it is unwinding in both the directions, and that's why it can be considered as bidirectional. Eukaryotes, that is the majority of the organism which have well developed nucleus. So in them, basically the DNA replication proceeds in both the directions, and that's why it is considered as bidirectional. Now dear students, once the two strands have separated, as you can see in this picture that the two DNA strands are separated, and we already have said that these two strands are separated by the enzyme DNA helicase, which actually breaks the hydrogen bonds between the two strands. So once the two strands have separated, now there are some special class of proteins. What are these proteins called? These proteins are known as SSBP proteins. What's the full form of SSBP? SSBP stands for single stranded binding protein. What's the role of this protein? Once the two strands have separated, now you can imagine that the two strands have separated, but once the two strands are separated, there is a high chance that the two strands again might coil. It can coil back. So there has to be some way by which the two strands should be prevented from again coiling, and that is majorly done by this SSBP. That's why these are known as single stranded binding proteins. That means these proteins are going to prevent the two strands from coiling back. Now once the two strands are separated, as you can see dear students, that the replication fork has been created. This is an inverted Y-shaped structure, right? Now please remember dear students, each of these separated strands will be considered as a template strand. Why is it called as a template? Template means these are the original strands, dear students, and on these strands actually the new DNA strands are going to be formed. So in this particular picture, as you can see, the blue strands which are getting formed, these are the new ones, whereas the orange strands which are there, these are the old strands. So the old strands are called as the template strands. Once the two strands are separated, as we said, the shape is looking like a replication fork. It is also considered as an inverted Y-shaped structure, and on this strands the new strands are going to be formed. Now if you consider this particular strand dear students, that is 5' to 3', on this strand a new strand is formed continuously. Now this strand which is formed in a continuous manner is considered as a leading strand, whereas on this particular strand, template strand, the DNA is formed in a discontinuous manner. This strand is considered as a lagging strand, right? So now see, please try and understand dear students over here, for the formation of leading strand or the lagging strand, right, there is a requirement of a small RNA molecule. Now as you can see in this picture, there is this red color thing which is attached at the 3' end. So this red color molecule, which is nothing but a small RNA molecule which is attached at the 3' end. What is that small RNA molecule called? This is known as the RNA primer. Now what's the role of RNA primer, guys? So RNA primer actually attracts the complementary nucleotides, or you can say it initiates the process of DNA. That means it starts the formation of new strand. It attracts the nucleotide towards itself, and that's how once it starts attracting the nucleotides towards itself, right, later on the DNA polymerase enzyme will start forming the new strand. The DNA polymerase enzyme will actually start forming the new strand. So as you can see in this picture, this enzyme called as DNA polymerase, this is the one which actually forms the new strand, and this new strand can be formed only if there is a primer which is attached at the 3' end. So on one side, as you can see, the DNA is formed in a continuous manner, and this is called as leading strand, whereas on the other side the DNA is formed in a discontinuous manner, which is in the form of fragments, right? So this strand is called as the lagging strand. So going ahead, as we can see that the strands, the new nucleotides keep on coming, and DNA polymerase will actually start forming the complementary strand. So when the complementary strand is formed, how does the complementary base pairing occur, guys? So always please remember, there is a rule which is called as complementary base pairing rule, which says that always the adenine will pair with thymine, whereas cytosine will pair with guanine. Such a rule is called as, or it is also known as complementary base pairing, wherein adenine and guanine, these are nothing but the purines, whereas cytosine and thymines are nothing but the pyrimidines. So always purines pair with the pyrimidines, and adenine, as you can see in this picture, adenine is pairing with thymine, cytosine is pairing with the guanine. Such a pairing is called as complementary base pairing rule or complementary base pairing. So as we said, dear students, that the DNA polymerase will now start forming the new strands, and this new strand which will be formed by adding the complementary nucleotides, right? So on one side the DNA is formed in a continuous manner, on the other side the DNA is formed in a discontinuous manner, right? Now when we say that the other strand which is formed in the discontinuous manner, it is in the formation of fragments, and these fragments are called as Okazaki fragments. Now why are they called as Okazaki fragments, dear students? Because these fragments were found out by a scientist whose name was Okazaki, whereas the other strand is formed in a continuous manner, right? So please remember that one strand, which is the leading strand, is moving in the direction of the fork, whereas the lagging strand is moving away from the. Now please remember, dear students, once the basically the two strands are being formed, that is, one is the lagging strand and other is the leading strand, but the primers are still there, so that RNA primer has to be removed, right? So there has to be some enzyme which is involved in the removal of primers. So please remember, in case of prokaryotes, the name of the enzyme is DNA polymerase 1, whereas in case of eukaryotes, the enzyme is called as DNA polymerase alpha. So these two enzymes help in removal of the RNA primers. Now finally, as you can see, guys, once the new strands are been formed, that is, one in a continuous manner, another is in the form of fragments. But now once the two strands are formed, again the strands have to be coiled back, and this is majorly done by the enzyme. As you can see on the screen, this enzyme is known as topoisomerase. Dear students, topoisomerase is also referred as DNA gyrase. So this DNA gyrase helps in coiling the DNA again. That is, the two strands will be coiled again, and once the two strands are coiled back, you can see now the two DNAs are formed. Now dear students, as you can see in this case, it is called as a semi-conservative DNA replication. Now why is it called as a semi-conservative DNA replication? Because in this DNA replication, on the original strand, as you can see, these are the two original strands. The ones which are in blue are the old strands. On this, the new DNA strand has been formed. That means when the two new DNA molecules are formed, half of the DNA is new, whereas half of the DNA is old. As you can see, it has been mentioned that there is an old strand in the new DNA molecule as well as there is a new strand. That means the 50% of the DNA which is formed is old DNA, and the 50% of the DNA is the new DNA. So in the new DNA, actually there is half of the DNA is old, half of the DNA is new, and such a type of DNA replication is called as semi-conservative DNA replication. So dear students, how was the topic? I hope you have understood DNA replication very well. So if you have understood the topic, please like, share and subscribe to One Page Biology. I will be coming with more videos in the coming days. Till that time, take care.